Usually when we talk about a medical diagnosis, there is this inherent expectation of precision. It feels almost akin to engineering, doesn't it?
Oh, absolutely. I mean, if you fall and break your arm, the radiograph shows a jagged white line across the radius, and the doctor simply points to the film and says, right, there it is. That is the problem.
Exactly. It's entirely binary. The bone is definitively broken or it is definitively intact. The causality is linear. The evidence is entirely visible. And, well, the treatment pathway is structurally obvious.
And there is immense comfort in that clarity. As clinicians, and frankly as patients too, we like our pathology to be visible. We want our illnesses categorized into these neat, discernible little boxes.
We really do. But the moment you step into the world of neurodevelopment or trauma or psychiatric illness generally, that radiograph machine is entirely useless. We are suddenly navigating a diagnostic landscape that is, quite frankly, incredibly murky. I mean, we cannot point to a single broken neuron on a scan and say, there is the depression.
Right, which forces a fundamental shift in how we approach the entire field of medicine.
Right.
We are forced to recognize that psychiatry is fundamentally the study of how brain, mind, and experience interact. And crucially, how that dynamic, continuous process can go wrong.
Yeah.
We are never just treating a brain floating in a jar, right? Nor are we treating abstract thoughts completely detached from biology. We are attempting to heal a deeply integrated, constantly adapting system.
And that interaction, that's really the core anchor for our deep dive today. So for the psychiatry trainees listening, the registrars, or just the fiercely curious clinicians joining us, our goal is to build a robust mental model of that integrated system with you.
Spot on.
We are acting as a companion guide to Chapter 18 of Kaplan and Sadok's comprehensive textbook of psychiatry. So, keep that text in mind because we are diving straight into the biological architecture of mental illness, specifically the genome, the transcriptome, and the proteome.
And to genuinely understand this architecture, we have to completely abandon that mid-20th century idea of genetic determinism. The notion that you have this static blueprint that dictates your destiny is not just outdated, it is biologically inaccurate.
Let's build a framework for this, using the metaphor of a massive, elaborate theatrical production, because I think that really helps. In this biological theater, the genome, the DNA you inherited, is the written script.
Right, the foundational text.
Exactly.
Yeah.
It contains all the potential dialogue and the stage directions. But a script sitting on a dusty desk is not a play, it's just paper.
Yeah, the text itself is inert. It requires interpretation, editing, and execution.
Precisely. So moving up a level, we encounter the transcriptome. If the genome is the script, the transcriptome is the director's interpretation and, say, the rehearsal process. It represents the active decisions being made in real time.
Which lines from the script are going to be read aloud today? Which scenes are going to be completely skipped because the audience is restless?
Right.
And the emotional timbre used to deliver the lines that actually remain.
Yeah, and finally, we have the proteome. The proteome is the live performance on opening night. It is the actors on stage, the functional molecules actually moving around the biological theater, carrying out the cellular processes, interacting with one another, responding to the lights and the crowd.
That framing is just so vital because it introduces the concept of multi-omics. Multi-omics is the integrated study of all these layers, genomics, transcriptomics, proteomics. It demonstrates why psychiatrists cannot simply look for a single broken line in the script to explain a profoundly complex phenotype like schizophrenia or bipolar disorder.
We are not hunting for a gene for depression.
Exactly. We are observing a dynamic interacting system where the script, the director, and the actors are constantly influencing one another, all while reacting to the environment and the patient's lived experience.
It completely reframes clinical assessment. You aren't just taking a history to find a static flaw in their code. You're trying to understand the live performance of their biology at this specific developmental stage. And, well, to appreciate why the field had to adopt this multi-layered view, we really should look back at the historical hubris that preceded it. I'm thinking of the late 1990s.
The great race to sequence the human genome.
Yeah.
It was an era of extraordinary optimism, but also profound biological arrogance, really. As we approach the year 2001, heading toward the publication of the first draft of the human sequence, the scientific community was quite literally placing wagers on how many genes we would find.
And the estimates were incredibly high. I've seen historical literature where researchers were confidently predicting 100,000 genes. Some estimates pushed as high as 160,000.
Which, logically, you can understand the reasoning.
Sure, we are Homo sapiens. We possess the cerebral cortex, which is arguably the most complex structure in the known universe. We compose symphonies, we launch satellites, we ponder our own mortality. The assumption was naturally that our instruction manual must be the most massive, complex genetic script in the entire animal kingdom.
Yeah, it's a very straightforward, linear assumption. More complexity in the organism must equal more genes in the code. But when the sequencing data finally materialized, 4 very curious patterns emerged that entirely dismantled that assumption.
Right.
The first shock was the raw, undeniable number. We did not have 160,000 protein coding genes. We barely had a fraction of that.
Staggering.
The current gene code annotation, which is constantly being refined, puts the total number of human protein coding genes at about 19,881. Let's call it roughly 20,000.
I want to pause on that number for you listening because the comparative biology is just baffling. Humans have roughly 20,000 protein coding genes. But if you look at a botanical source, a simple fern has 37,500 genes. And a single-celled organism, Amoeba dubia, boasts roughly 50,000 genes housed in a genome that is literally 200 times larger than ours. So how does a single cell require a script 200 times longer than the organism that invented the radiograph machine?
Well, that amoeba paradox highlights the second and third patterns revealed by the sequencing project. Gene number does not predict organismal complexity. Furthermore, raw genome size does not predict complexity.
Right.
To put it in mammalian terms, mice and humans have roughly the exact same number of protein coding regions. Humans and chimpanzees share essentially the same size genome. The raw material is remarkably similar.
So relying on the size of the script to measure complexity is like assuming a 1,000-page novel is inherently more sophisticated than a 100-page poem simply because it weighs more.
That's a brilliant way to put it. The amoeba might be a genetic hoarder, holding on to hundreds of billions of base pairs of repetitive junk, while human biology is doing something entirely different with a much smaller toolkit.
Precisely. The critical question for a neuroscientist or a psychiatrist then becomes, if our genome is not particularly distinctive in its size or its gene count, how did it possibly produce the unique, staggering qualities of the human central nervous system?
And the answer to that lies in the fourth and perhaps most humbling pattern discovered by the project. Of those 3 billion base pairs in our DNA, a shockingly minuscule percentage actually codes for proteins, is roughly 2%.
2%, meaning 98% of the human genome is non-coding.
Yeah. And for decades, that 98% was dismissively labeled junk DNA. The prevailing thought was that if a sequence did not directly translate into a functional protein, if it didn't produce an actor for the stage, it was merely evolutionary baggage.
Just a fossil record of extinct viruses and ancient mutations that we dragged along for the ride.
Exactly. But the textbook makes it very clear that this so-called junk is actually the genomic dark matter. It is the very foundation of human complexity.
Because the magic of human neurobiology is not in possessing more genes. The magic lies in the incredibly sophisticated, highly regulated ways we slice, interpret, and manage the 20,000 genes we do have.
Which brings us straight back to the director in your theater metaphor. The 2% represents the actors. The 98% represents the directors, the stage managers, the lighting cues, and the architectural scaffolding of the theater itself.
It's everything that makes the play actually happen.
Yes. And to understand how that 98% regulates the system, we have to look at the physical packaging of the DNA. Because in biology, physical structure dictates function.
Let's visualize this physical structure for a moment. We're talking about 3 billion nucleotides, the A, C, T, and G base pairs. If you were to extract the DNA from a single human cell and stretch it out linearly, it would be, approximately 2 meters long?
Roughly 2 meters, yes.
Yet that entire 2-meter thread has to be compacted into a cell nucleus that is measured in micrometers. It is microscopically small.
The logistical challenge is immense. To manage this, the cell employs specialized proteins called histones. You can visualize histones as microscopic spools. The 2-meter thread of DNA winds tightly around these histone spools to form repeating complexes known as nucleosomes. These nucleosomes then coil and stack upon each other to create a dense superstructure called chromatin.
So chromatin is the organized configuration of the thread on the spools. But it isn't just a static ball of yarn, is it? The way it is packed determines whether the genes inside it can actually be read.
Exactly. When researchers look at chromatin under a powerful microscope, they see distinct regions of light and dark banding. The lighter, less densely packed material is called euchromatin.
And because the DNA here is relatively open and unschooled, the cellular machinery, the enzymes that transcribe the script, can physically access the genetic code.
Yes. Euchromatin is the active state. It is where the genes are turned on.
Right. And the darker, denser material is heterochromatin. It is wound so tightly that the transcription machinery simply bounces off. The genes trapped inside heterochromatin are physically inaccessible, which means they are effectively silenced.
This physical winding and unwinding is the first massive layer of biological regulation.
Yeah.
By simply tightening the spools in one area and loosening them in another, a neuron can completely alter its functional state without changing a single letter of the underlying genetic code.
It's amazing, and this physical structure explains part of that 98%. The genome needs vast stretches of sequence just to handle the structural engineering of the chromatin. But the non-coding DNA is doing much more than just acting as scaffolding, isn't it? It is producing its own massive array of transcripts.
Oh, absolutely. The textbook details an entire universe of non-coding RNAs, or ncRNAs. We have currently cataloged over 30,000 non-coding genes. It's a fascinating trend in molecular biology right now. While the official count of protein-coding genes has actually decreased slightly as our annotations become more stringent, the number of recognized non-coding RNAs is absolutely skyrocketing. They act as the hidden puppet masters of the cell.
The sources outline a few specific categories of these puppet masters that are highly relevant to psychiatric research. Let's start with small interfering RNAs, or siRNAs. How do these short strands of RNA control the system?
Well, siRNAs are highly targeted regulatory molecules. They're short RNA transcripts designed to be perfectly complementary to a specific messenger RNA that has just been transcribed and is on its way to the ribosome to create a protein.
Okay.
Because the siRNA is a perfect mirror image of the target mRNA, it physically binds to it.
Creating a double-stranded RNA molecule, which, if I recall my basic virology, cells absolutely hate because it looks like a viral infection.
Exactly. The cell views double-stranded RNA as a threat or a critical error. When the siRNA binds to its target, it recruits a complex of proteins that acts like a pair of molecular scissors, slicing the target mRNA to pieces. The transcript is destroyed before it can ever produce a protein.
So in our theater, the protein-coding gene is the actor walking toward the stage, script in hand. The siRNA is the stage manager who steps out of the shadows, whispers, "You've been cut from this scene," snatches the script, tears it up, and pushes the actor out the back door.
That is a perfect translation of the mechanism. The gene has been silenced, but the original DNA in the nucleus was never touched.
Brilliant.
Then we have a closely related category called microRNAs, or miRNAs. They operate on a similar principle, but with a vital difference in severity.
How so?
miRNAs typically bind to what is called the 3' untranslated region, essentially the tail end of an mRNA transcript. Instead of triggering the total destruction of the message like the scissors do, they create a physical roadblock. They decrease the efficiency of the translation process.
Ah, I see. So siRNAs are an on-off switch, whereas miRNAs function more like a volume dial, just turning down the overall concentration of a specific protein in the neuron.
Yes, and crucially, a single miRNA can target hundreds of different mRNA transcripts, allowing it to turn down the volume on entire biochemical pathways simultaneously.
Now, here's a concept from the text that I found truly elegant. It involves pseudogenes and something termed the sponge hypothesis. The prefix pseudo obviously implies they are fake. What exactly is a pseudogene?
Pseudogenes are essentially genomic fossils. Over millions of years of evolution, genes occasionally duplicate. Sometimes one of those duplicated copies accumulates random mutations that destroy its ability to code for a functional protein. It becomes a broken relic of a gene.
Right.
The human genome contains over 14,000 of these pseudogenes. And historically, scientists assumed they were completely dead, transcriptionally silent junk.
But they aren't silent. They are actively transcribed into RNA. Why would a highly efficient human cell waste precious ATP cellular energy transcribing a fossil that cannot make a protein?
This is the sheer brilliance of the sponge hypothesis. Because a pseudogene shares a deep evolutionary history with its functional counterpart, its DNA sequence is still very similar. Consequently, the RNA transcript it produces is also very similar.
Okay.
This means the pseudogene RNA can bind to the exact same regulatory microRNAs that target the functional gene.
Ah. So it acts as a molecular decoy. If the cell is flooded with inhibitory miRNAs trying to turn down the volume on a vital protein, the pseudogene produces a massive amount of its own broken RNA. Those transcripts act like sponges, soaking up all the inhibitory miRNAs in the cytoplasm.
It is a form of molecular altruism, really. The pseudogene sacrifices its own transcripts to the regulatory machinery so that the true mRNA can slip past unscathed and produce its protein. It profoundly regulates the entire network without possessing any intrinsic protein coding function of its own.
That level of indirect regulation is just staggering. And it scales up too. We aren't just dealing with tiny short RNAs. The text also details long intergenic non-coding RNAs, or LINCs. These are massive transcripts.
LINCs are fascinating because they can exert influence on an entire chromosome. The classic example here is the SST RNA. This is responsible for X chromosome inactivation in human females.
Right.
Because females possess 2 X chromosomes and males possess only 1, there needs to be a mechanism to ensure females do not produce double the required amount of X-linked proteins.
Dosage compensation.
Correct. The SST gene does not make a protein. Instead, it produces a massive LINC RNA transcript that physically coats one entire X chromosome. This coding triggers a cascade of epigenetic changes that compress that specific chromosome into dense, silent heterochromatin.
It shuts down almost an entire chromosome using only a non-coding RNA.
Exactly.
It is remarkable how much heavy lifting the drink DNA is actually doing. But we have to address what is arguably the most disruptive revelation regarding the non-coding genome. I'm referring to repetitive DNA, and specifically the jumping genes.
Transposable elements, or transposons, yes. This is where the concept of a static, unyielding genome completely falls apart. Nearly 1/3 of the entire human genome consists of repeated elements. But the most ubiquitous and perhaps the most relevant to neurobiology are the ALU elements.
I read the section on ALU elements multiple times to ensure I was understanding the implications correctly. What exactly is an ALU element structurally?
An ALU element is a relatively short stretch of DNA, typically about 300 base pairs long. It belongs to a classification known as short interspersed elements, or SCEs. What makes an ALU element extraordinary is that it is a retrotransposon.
Meaning?
It possesses the biological machinery to copy and paste itself. It transcribes its own sequence into RNA, utilizes a specialized enzyme to reverse transcribe that RNA back into DNA, and then physically inserts that new DNA copy into a completely random, novel location within the genome.
And according to the textbook, these jumping elements make up 10% of our entire genome. A single self-replicating sequence occupies 1/10 of our genetic code. How long has this been happening?
This is a deeply ancient primate phenomenon. We can trace massive waves of these insertions through our evolutionary histoy. The ancient AluJ subfamily swept through the genome around 81 million years ago. The Alu subfamily expanded about 48 million years ago.
Right.
But the most critical for us is the young AluY wave. This wave began a mere 6 million years ago, right around the time our lineage diverged from chimpanzees. And the astonishing fact is that the AluY elements are still active today. There are over 1 million copies of ALU elements sitting in your genome right now.
Now here's the revelation that forces a complete paradigm shift. The text explicitly states that these ALU elements are actively transposing, actively jumping and inserting themselves in human neurons in vivo. This is happening right now, inside the brains of everyone listening to this discussion.
Yes, for decades, the central dogma of genetics was stability. We assumed that the DNA you inherited at conception was meticulously preserved, copied identically into every single cell of your body, and remained static until your death. But the discovery of somatic retrotransposition in the brain shatters that illusion. In your cerebral cortex, these ALU elements are actively waking up, making copies of themselves, and inserting those copies into new locations within individual mature neurons.
The phrase that comes to mind is genetic jazz. The DNA in our neurons is improvising. Every time an ALU element jumps and lands in a new location, it fundamentally alters the sequence of that specific neuron.
Precisely.
It might land in the middle of a promoter, changing how a gene is expressed. It might land in an intron and alter how the RNA is spliced. It means that my brain is not governed by one singular genome. My brain is a mosaic composed of billions of slightly different, genetically unique neurons.
And if we extrapolate that to psychiatric vulnerability, the implications are profound. If we discover that the rate of this transposition is influenced by environmental factors, say by early childhood trauma, chronic stress, or even learning and memory formation, it implies that our lived experiences might actually be subtly rewriting the genetic code of our individual neurons over the course of our lifetime.
The environment isn't just interacting with the genome, it may be actively rearranging it.
Exactly.
It fundamentally blurs the line between nature and nurture. If the nurture physically alters the nature, the dichotomy collapses. And, speaking of ancient organisms altering our biology, we must recognize that we don't just possess one genome in our cells, do we? We have to factor in the mitochondrial genome.
We must. This traces back to the endosymbiotic theory. Approximately 1.5 to 2 billion years ago, a primitive eukaryotic cell essentially engulfed a free-living, oxygen-consuming bacterium, but rather than digesting it, the host cell and the bacterium entered into a mutually beneficial symbiotic relationship.
And that ancient bacterium eventually evolved into the mitochondria, the organelles responsible for generating the vast majority of our cellular energy in the form of ATP.
Right, and because of that distinct evolutionary origin, mitochondria possess their own separate circular DNA, completely distinct from the human DNA in the nucleus.
But over the course of 2 billion years, those two genomes haven't remained entirely isolated. There has been a steady migration of genetic material.
A very significant migration. The human nuclear genome and the mitochondrial genome are now deeply intertwined. Over evolutionary time, nearly 300 genes that originally belonged to the mitochondria have physically migrated and integrated themselves into our 23 human chromosomes. We refer to these as NUMTs, or nuclear DNA of mitochondrial origin.
What evolutionary advantage does the cell gain by moving mitochondrial genes into the main human nucleus? Why not just leave them where they are used?
It is almost certainly a protective strategy. The mitochondria are the powerhouses of the cell, but generating all that ATP is a messy, highly oxidative process. It creates reactive oxygen species-free radicals that act like molecular shrapnel, severely damaging nearby DNA.
Ah, I see.
The mitochondrial genome is highly vulnerable because it sits right next to the furnace and it lacks the protective histone spools we discussed earlier. By migrating the most critical mitochondrial genes into the main nucleus, where the DNA is heavily shielded by histones and robust repair mechanisms, the cell ensures that the vital instructions for energy production are not destroyed by the very energy they create.
It is a brilliant evolutionary safeguard. And the textbook notes that this is an ongoing process. 27 of these NUMTs were incorporated into the human genome within just the last 4 to 6 million years. They are so recent that they aren't even present in chimpanzees.
Exactly. This ancient bacterial DNA is actively shaping human evolution.
But bringing this back to the clinic, why does a psychiatrist need to care about mitochondrial DNA?
Because mitochondrial function and the energy it provides is the absolute bottleneck for all the complex genomic regulation we're discussing. Jumping genes, alternative splicing, chromatin remodeling. All of this requires massive amounts of ATP. Furthermore, mitochondrial processes are now recognized as a central hub for the body's response to systemic stress.
The text mentions a multi-omics study conducted on astronauts, actually.
Yes, spaceflight is a model of profound, multifaceted stress radiation, microgravity, psychological isolation. When researchers analyzed the multi-omics data of astronauts, they found that mitochondrial stress was the central biological node reacting to that environment.
That's fascinating.
In psychiatry, mitochondrial dysfunction is increasingly implicated in the pathophysiology of chronic stress disorders, major depression, and neurodegeneration. When a patient endures profound trauma, that psychological experience translates into a metabolic demand that physically stresses this ancient energy system.
And if the mitochondria in one area of the brain are feeling under stress, how does that distressed cell communicate its state to the rest of the neural network? The biological postal service mentioned in the literature involves exosomes.
Exosomes are a critical frontier in neuroscience. They are a type of extracellular vesicle-tiny, membrane-bound lipid spheres secreted by cells. For a very long time, the consensus was that these vesicles were essentially cellular garbage bags, a mechanism for the neuron to expel waste products into the extracellular space.
But the reality is far more complex.
Much more. We now understand that exosomes are highly curated cargo ships. They contain specific sequences of DNA, regulatory RNAs like the miRNAs we discussed, and functional proteins. They represent a sophisticated method of intercellular communication.
So a neuron in the prefrontal cortex can package a specific regulatory microRNA into an exosome, secrete it, and fundamentally alter the transcription of a neighboring glial cell.
Exactly. And crucially, these exosomes can cross the blood-brain barrier. This has immense implications for neurodegenerative diseases like Alzheimer's or Parkinson's. In neurodegeneration, exosomes exhibit a deeply paradoxical nature.
How do you mean?
On one hand, if a neuron is overwhelmed with toxic misfolded proteins, it can package those aggregates into an exosome and expel them. This local action saves that individual neuron from apoptosis.
But the hot potato has to go somewhere.
Exactly. If that toxic exosome is subsequently taken up by a neighboring healthy neuron, the misfolded proteins can seed aggregation in the new cell. The exosome becomes the vector for spreading the pathology across the brain network.
While that sounds devastating, understanding that mechanism also provides an extraordinary therapeutic opportunity.
It really does, because exosomes naturally cross the blood-brain barrier, a barrier that blocks roughly 98% of all small molecule drugs. Bioengineers are intensely focused on them. The future of psychiatric and neurological treatment may involve harvesting exosomes, emptying their contents, and loading them with specific synthetic siRNAs designed to silence a mutant gene or with neurotrophic factors.
We could potentially deliver highly targeted multi-omics therapies directly into the brain parenchyma without requiring invasive neurosurgery.
That's the hope.
It is a breathtaking prospect. All right, let's transition from the structural complexities of the genome and look closely at the transcriptome. This is the rehearsal space in our metaphor. We established earlier that the human genome only has about 20,000 protein-coding genes. How does the cell generate the infinite variety required to build a human brain from such a limited script?
The primary mechanism driving that variety is RNA splicing. To understand splicing, we have to look at the anatomy of a gene. When a gene is first transcribed from DNA into a raw strand of RNA, that transcript contains two distinct types of sequences. It contains exons, which are the actual coding regions that translate into amino acids, and it contains introns, which are long, non-coding intervening sequences.
Returning to our theater metaphor, the exons are the actual lines of dialogue the actors must speak. The introns are the director's lengthy, rambling notes scrawled in the margins between the lines. You cannot read the script aloud with the notes included. It would be nonsensical.
Precisely. Before that raw RNA can be sent to the ribosome to be translated into a functional protein, those introns must be removed. The cell uses a massive molecular machine called the spliceosome. The spliceosome physically cuts out every single intron and meticulously splices the remaining exons together into a continuous, mature messenger RNA.
But the profound trick of human biology is that the spliceosome does not always splice the script the exact same way every time.
No, and that is the concept of alternative splicing. Imagine a single gene that contains 10 distinct exons. In a liver cell, the spliceosome might include all 10 exons in the final mRNA. But in a neuron, under specific physiological conditions, the spliceosome might decide to cut out exon number 3 and exon number 7, splicing exon 2 directly to exon 4.
And the resulting mRNA sequence is completely different, which means the final protein it produces will fold into a different three-dimensional shape and potentially serve a completely different function, all originating from the exact same stretch of DNA.
The scale of this process is what separates us from the amoeba. The textbook highlights a staggering statistic. Over 80% of all human protein-coding genes undergo alternative splicing. And the single tissue in the human body that exhibits the highest rate and complexity of alternative splicing is the brain.
Which elegantly resolves the paradox of our relatively small gene count. We don't need 100,000 separate genes because we can endlessly slice and combine our 20,000 genes to create hundreds of thousands of distinct protein variants.
Furthermore, alternative splicing provides a massive evolutionary shortcut. It creates what geneticists call a neutral space for evolution. Let's say a specific gene performs a function that is absolutely critical for basic cellular survival. If a random mutation occurs in that gene and destroys its function, the organism dies.
Right.
That represents incredibly strong negative selection pressure. Evolution cannot experiment with that gene.
It is entirely locked down.
But alternative splicing circumvents this. A critical gene will typically have major use exons, the core components that are almost always included in every transcript. These major exons are highly conserved across millions of years. But the gene will also have minor use exons, which are only occasionally spliced into the final product.
So the cell can safely tolerate random mutations occurring within those minor use exons?
Exactly. Because even if a mutation in a minor exon produces a flawed protein, the cell is still producing the critical, life-saving version of the protein most of the time via the major exons. The organism survives.
That's incredibly clever.
It allows the brain to safely test out new, slightly altered versions of a receptor or an enzyme in the background. If one of those newly mutated minor exons happens to create a dopamine receptor with a slightly more efficient binding affinity, it provides a subtle evolutionary advantage and slowly becomes fixed in the population. It is biological beta testing without risking catastrophic system failure.
The director is running alternative scenes and rehearsals just to see what works with the audience without ever ruining the core narrative of the play. It's brilliant. But alternative splicing is just reordering the existing words. The text introduces another mechanism called RNA editing, and I have to admit, as a clinician, this mechanism feels almost dangerous. Walk us through ADAR-mediated RNA editing.
RNA editing is truly one of the most remarkable post-transcriptional modifications. In standard molecular biology, the sequence of the mature RNA is expected to be a perfect, faithful copy of the DNA sequence it was transcribed from. But in RNA editing, a specialized family of enzymes known as ADARs physically locate the RNA transcript and chemically alter the sequence after it has been transcribed.
Wait, hold on. The script is locked in the DNA. The transcription machinery makes a perfect copy. But then this ADAR enzyme acts like a rogue director with a red pen, just crossing out letters and writing in new ones before handing the script to the actors.
Basically, yes.
Doesn't that risk total biological chaos? How does the cell prevent the enzyme from just mutating critical messages into nonsense or promoting oncogenesis?
Well, it is a highly regulated risk.
Yeah.
ADAR enzymes do not edit randomly. They recognize specific secondary structures in the RNA. The mechanism itself is a deamination reaction. The ADAR enzyme locates a specific adenosine and A nucleotide on the RNA strand. It chemically removes an amino group, converting that adenosine into a different molecule called inosine, or I.
And why does an I matter to the cell?
Because when the cellular machinery, the ribosome, reads that transcript, it does not recognize inosine as an adenosine. It reads inosine as if it were a guanosine, a G.
Right.
So functionally, an A has been magically converted into a G. If this edit occurs within a coding exon, it changes the 3-letter codon. Changing the codon changes the amino acid that gets incorporated. Changing the amino acid fundamentally alters the physical properties of the final protein. All of this occurs without a single mutation in the underlying genomic DNA.
And where in the human body is this rogue editing happening most frequently?
In the central nervous system, humans exhibit the highest levels of A to I RNA editing of any primate, and it is heavily concentrated in transcripts that code for crucial neurological hardware. We see extensive editing in AMPA receptors, which mediate fast excitatory transmission, in serotonin receptors, in GABA receptors, and in various voltage-gated ion channels. It acts as an intricate fine-tuner of the brain's neurophysiology.
The clinical implications of this are just staggering. If I have a psychiatric patient who is exhibiting severe dysregulation in their serotonergic system, a traditional genetic screen might show that the DNA sequence for their serotonin receptors is perfectly normal. There is no mutation. Yet the malfunction could be occurring entirely in the editing room, where the ADAR enzymes are failing to edit or hyper-editing the RNA transcripts in response to some environmental trigger.
This represents the frontier of psychiatric research. We are moving away from merely cataloging DNA mutations and moving toward understanding the dynamic regulation of the transcriptome, which seamlessly brings us to the ultimate intersection of environment and genetics, epigenetics.
Epigenetics. The classic definition is heritable changes in gene expression that do not involve any alteration to the underlying nucleotide sequence. It is the system of chemical tags physically placed on top of the DNA and the histones. Let's break down the mechanics of the two primary epigenetic systems. First, DNA methylation.
DNA methylation is relatively straightforward. Specific enzymes, called DNA methyltransferases, attach a tiny chemical molecule, a methyl group, which is just 1 carbon and 3 hydrogens, directly onto a cytosine base in the DNA. This almost exclusively occurs at what we call CpG sites, where a cytosine sits immediately adjacent to a guanine.
What happens when a region of DNA, particularly the promoter region where transcription begins, accumulates a heavy concentration of these methyl tags?
It creates a profound physical barrier. The transcription factors and RNA polymerase simply cannot bind to the DNA because the methyl groups are physically in the way. Heavy DNA methylation at a promoter is a hallmark of gene silencing. It firmly locks the gene in the off position.
It is analogous to placing a heavy padlock on that specific scene of the script. The pages are still there, but no one can open them to read the words.
Exactly. But crucially, these padlocks are not permanent. The epigenetic landscape is dynamic. The methyl group can be enzymatically oxidized into a variant called 5-hydroxymethylcytosine, or 5-HMERC. The conversion to 5-HMERC is essentially the first step in removing the padlock and turning the gene back on.
Oh, interesting.
Interestingly, 5-HMERC is found in remarkably high concentrations in the mammalian central nervous system, and recent research indicates it plays novel roles in regulating how genes are spliced in the brain.
So the brain is constantly adding and removing these padlocks. The second major system involves the histones, those molecular spools we discussed earlier. The spools themselves get tagged, correct?
Yes, the histone proteins have long, flexible molecular tails that hang off the main spool. These tails are rich in specific amino acids, primarily lysine, which is denoted in biochemistry by the letter K. The cell can add various chemical tags, methyl groups, acetyl groups, to these specific lysines.
Now, I want to clarify how a clinician or researcher reads these tags because the nomenclature in the literature can look like an impenetrable alphanumeric code. If a researcher is reading a paper and sees a tag denoted as, say, H3K27ME3, what is the underlying mechanism they should visualize?
They should visualize a specific address on the spool. H3 tells you it is histone number 3. K27 tells you the tag is attached to the 27th amino acid on that tail, which is lysine. And Me3 tells you that 3 methyl groups have been attached.
Okay, that makes sense.
While the exact code is complex, the functional implication is what really matters. When a researcher sees heavy trimethylation at H3K27 or at H3K9, that combination acts as a massive molecular stop sign. It chemically forces the chromatin to wind tightly into dense heterochromatin. The gene is violently repressed.
So 9 and 27 are the stop signs. How does the cell signal a green light?
Different addresses. If the cell attaches methyl groups to H3K4 at the promoter, or H3K36 along the body of the gene, those specific tags repel the repressor proteins. They force the chromatin to unspool into open active euchromatin. That gene is actively being transcribed.
Why is this mechanical understanding so vital for psychiatry? How does the placement of a microscopic methyl group alter our understanding of clinical pathology?
Because epigenetics provides the biological mechanism for phenomena that raw genetics simply cannot explain. Consider monozygotic twins. They possess identical genomic DNA. Yet, in the context of a complex disorder like schizophrenia, the concordance rate between identical twins is roughly 50%, not 100%.
Right.
If one twin develops the disorder, the other twin still basically has a coin flip chance of developing it, despite having the exact same genetic code.
The traditional explanation was always vaguely attributed to, the environment, but epigenetics provides the literal bridge. The twins experience different environmental stressors, perhaps a viral infection in utero, different nutritional environments, or disparate psychological traumas later in life.
Exactly.
Those different environmental inputs triggered enzymes to place different epigenetic tags, different padlocks, and stop signs on their identical DNA, silencing completely different networks of genes.
It beautifully illustrates that vulnerability is not just inherited, it is cultivated. Epigenetics also provides the mechanism for genomic imprinting. This is a fascinating phenomenon where the expression of a gene depends entirely on which parent you inherited it from.
The classic pediatric examples are Prader-Willi syndrome and Angelman syndrome. Walk us through how epigenetics drives those distinct phenotypes.
Both Prader-Willi and Angelman syndromes can be caused by a physical deletion of the exact same stretch of DNA on chromosome 15. The genetic mutation is identical. However, the resulting clinical presentation is vastly different. Prader-Willi presents with profound hyperphagia, obesity, and mild intellectual disability. Angelman syndrome presents with severe intellectual disability, ataxia, and a characteristic happy, excitable demeanor.
How can the exact same missing sequence produce two entirely different syndromes?
Because the genes in that specific region of chromosome 15 are epigenetically imprinted. In a healthy individual, certain genes in that region are permanently padlocked, methylated on the chromosome inherited from the mother, while different genes are padlocked on the chromosome inherited from the father. If a child inherits the deletion from their father, they lose the paternal genes, and because the maternal copies are naturally epigenetically silenced, they have 0 functional copies of those specific genes, resulting in Prader-Willi. If they inherit the identical deletion from their mother, they lose the maternal genes, and the natural silencing of the paternal copies results in Angelman syndrome.
The epigenome literally remembers the parent of origin. The textbook also explicitly links parent of origin effects to psychiatric risk, specifically regarding autism spectrum disorder.
Yes. In the same region of chromosome 15, we sometimes see duplications rather than deletions. If a child inherits a duplicated segment of 15q from their mother, it significantly increases their risk of developing autism spectrum disorder. However, if they inherit that exact same duplication from their father, the risk profile is vastly different and often benign. The gender of the transmitting parent dictates the epigenetic state of the chromatin, which fundamentally alters the neurodevelopmental trajectory of the child.
That is a profound layer of regulation. And just when I thought the tagging system was exhaustive, the text introduces the epitranscriptome. So we can methylate the DNA, we can tag the histone spools, and apparently the cell can also place chemical tags directly onto the RNA messenger itself.
The layers of regulation are truly fractal. The epitranscriptome refers to biochemical modifications of the RNA. The most abundant and well-studied of these modifications is M6A N6-methyladenosine. Enzymes attach a methyl group to an adenosine base within the mRNA transcript.
What does tagging the RNA achieve that editing or splicing doesn't?
It controls the logistics of the transcript. An M6A tag can act as a shipping label, dictating how quickly the RNA is exported from the nucleus to the cytoplasm. It can act as a timer, dictating how rapidly the RNA is degraded by cellular scavengers. Or it can determine how efficiently the ribosome translates the RNA into protein. Notably, the M6A modification is heavily implicated in the regulation of circadian rhythms. It governs the decay rate of transcripts for core clock genes.
The speed at which clock gene RNA degrades dictates the ticking of the biological clock. And considering how central circadian disruption is to the pathophysiology of bipolar disorder, major depression, and schizophrenia, the epitranscriptome might be a massive hidden variable in psychiatric illness. All right, we have navigated the genome and the transcriptome. Let's examine the final output, the proteome, the live performance on opening night.
One might assume that once the RNA is finally translated into a protein by the ribosome, the regulation is complete. But proteins are rarely functional immediately upon synthesis. They must undergo post-translational modifications.
The actors have to put on their costumes and makeup before they walk on stage.
Exactly. The cell can fundamentally alter a protein's function through chemistry. Proteins can be physically cleaved in half to activate them. They can undergo glycosylation, or complex sugar molecules are attached to aid in folding or cell-to-cell recognition. They can be phosphorylated by kinases, which often acts as an incredibly fast on-off switch for enzymes. And they can be ubiquitinated. Ubiquitin is a small regulatory protein that, when attached to a target protein, acts as a cellular kiss of death. It flags the protein for immediate destruction by the proteasome.
The textbook uses the dopamine transporter, or DAT, to illustrate the critical nature of these modifications.
Yes, the dopamine transporter is essential for clearing dopamine from the synaptic cleft, thereby terminating the dopaminergic signal. When the DAT protein is synthesized, it is essentially inert. It must undergo extensive post-translational modifications, including highly specific glycosylation and phosphorylation events, before it can be correctly routed to the plasma membrane of the synapse and function efficiently.
And if that fails?
If those post-translational modifications are flawed due to cellular stress or genetic variation, the transporter fails. Dopamine pools in the synapse, leading to massive alterations in reward circuitry and motor control, which are core features of numerous psychiatric and neurological conditions.
Now, I want to pause and synthesize everything we have covered, because the sheer scale of the maths involved in this system is difficult to conceptualize. The textbook provides a calculation for the complexity of the human proteome that forced me to reconsider how I view biological networks entirely. Let's walk the listener through this mathematical explosion, starting from the foundation.
It is a humbling exercise. We begin at the bottom layer, the genome. We possess approximately 3 billion base pairs of DNA. That sequence yields roughly 20,000 to 25,000 protein-coding genes.
25,000 variables. It is a large number, but it feels manageable for a supercomputer.
But then we introduce the transcriptome. We introduce alternative splicing. The textbook highlights the gene that codes for titin, a massive structural protein. The titin gene contains 234 exons. If you apply the combinatorial mathematics of alternative splicing, calculating all the possible ways you could include or exclude those 234 exons, that single gene could theoretically be spliced into 2.76 times 10 to the 70th power-distinct mRNA transcripts.
I just want to repeat that for emphasis. One single gene, 10 to the 70th power potential variations.
Yes. Now, extrapolate that across the entire genome. Factor in the 8' RNA editing. Factor in the epigenetic silencing. Factor in the vast array of post-translational modifications, cleavage, glycosylation, phosphorylation.
Right.
When you integrate all of these regulatory layers, biochemists estimate that the human body is capable of producing several million distinct functional proteins from those original 25,000 genes. Let's use conservative estimates and say the human proteome consists of 2 million distinct protein variants.
2 million unique actors on the biological stage.
But those actors do not stand in isolation. Biology is defined by interaction. Enzymes bind to substrates, receptors bind to ligands, structural proteins interlock. If you map out those 2 million proteins and calculate all their possible physical connections and signaling pathways, there are roughly 4 trillion possible protein interactions. And when you calculate the total theoretical states of this interacting network, the number approaches 7.0 times 10 to the 82nd power.
7 times 10 to the 82nd power. That number is so large it loses meaning without context.
Well, for context, theoretical physicists estimate that the total number of atoms in the entire observable universe is roughly 4.0 times 10 to the 80th power.
The mathematical permutations of the biological network inside a single human patient are greater than the number of atoms in the universe.
This is the definitive death of the broken switch model in psychiatry. We will never find a singular, isolated broken gear that causes schizophrenia. We're attempting to identify maladaptive patterns within a dynamic, constantly adapting, recursively interacting network that defies human comprehension.
Which brings us to the profound clinical challenge. How does all of this mind-bending infinite variation actually manifest in human disease? If the network is larger than the universe, how do we as clinicians ever identify the vulnerability when a patient walks into our clinic? How do we find the signal in the noise?
We begin by looking for variations that are shared across large populations. We have to categorize the types of genetic variation. The most common form is sequence variation, specifically the single nucleotide polymorphism, or SNP.
A SNP is the simplest possible change. It is a single-letter typo in the 3 billion-letter script. An adenosine is swapped for a thymine.
Correct. And the definition is important. To be classified clinically as a polymorphism rather than a rare mutation, that specific single-letter change must occur in more than 1% of the general population. We have sequenced thousands of human genomes and identified tens of millions of these common SNPs.
But if they are common, occurring in huge swaths of the healthy population, how do they cause disease? If I run a genetic test and find out a patient has a specific SNP in their dopamine receptor gene, does that mean they will develop schizophrenia?
Absolutely not. And understanding why requires understanding the common variant, common disease hypothesis. Historically, genetics focused on Mendelian disorders like Huntington's disease or cystic fibrosis, where a single, highly penetrant mutation unequivocally causes the disease. But psychiatric illnesses are highly heritable, complex traits.
Right.
When researchers began running genome-wide association studies, or GWAS, scanning the genomes of tens of thousands of psychiatric patients, the results were initially incredibly frustrating. They did not find a schizophrenia gene.
They found thousands of tiny signals scattered across the entire genome.
Exactly. The GWAS data revealed that disorders like schizophrenia, severe major depressive disorder, and bipolar disorder result from the combined cumulative effect of hundreds or even thousands of these common SNPs.
It's the bucket analogy. Each individual SMP is like a single drop of water falling into a bucket. One drop is meaningless. It confers a microscopically small increase in risk, perhaps raising the relative risk by a fraction of a percent. But if a patient unluckily inherits 500 of these specific, slightly deleterious drops from their parents, their genetic bucket is sitting dangerously full.
That polygenic risk score creates the vulnerability. But the illness still requires a trigger. The bucket is full, but it requires a profound environmental stressor, the trauma, the substance use, the viral infection, to finally kick the bucket over. That is when the biological threshold is crossed, the network fails to compensate, and the clinical phenotype of the illness manifests.
So that covers sequence variation, the single-letter typos. But what about structural variation? Because this concept fundamentally alters how we view the physical integrity of the genome. We aren't just swapping individual letters. Entire paragraphs of the script are missing.
We were referring to copy number variations, or CNVs. This is where large physical chunks of DNA sequences spanning thousands or even millions of base pairs are structurally altered. They can be duplicated, leaving the patient with 3 or 4 copies of a gene instead of the standard 2. Or they can be deleted entirely, leaving the patient with only 1 copy or a complete absence of that genetic sequence.
The shocking revelation in the literature is that these massive structural alterations are not inherently lethal. In fact, healthy individuals are walking around with them right now. I could be missing 50 large chunks of my DNA and possess extra copies of 50 others and exhibit absolutely no pathological phenotype.
Yes, the average healthy human carries dozens, if not hundreds, of these CNVs. It was a massive surprise to the genetic community. It demonstrates that the human genome tolerates haploinsufficiency, the state of possessing only 1 functional copy of a gene, far better than we historically believed. The network is incredibly resilient.
I must push back on this concept on behalf of the clinicians listening, though. If we accept that healthy people are constantly walking around missing entire chunks of DNA, acting as harmless genetic background noise, how on earth does a psychiatrist or a pediatrician looking at a genetic microarray know if a deleted gene is the cause of a patient's neurodevelopmental delay, or if it is just a coincidental, benign variation?
That question represents the defining challenge of modern medical genetics. The days of simply spotting a deletion on a test and declaring, we found the cause, are over. It requires rigorous clinical reasoning and immense databases of population genetics.
So it's about context.
Yes, the clinician must evaluate the type and the frequency of the CNV. If the deletion is seen in 5% of the healthy population, it is likely just another drop in the polygenic bucket. However, if the CNV is exceptionally rare, or critically, if it is de novo, the clinical index of suspicion rises dramatically.
Let's define de novo clearly. It means new. The mutation is not present in the somatic cells of the mother or the father. It occurred spontaneously during the formation of the sperm or the egg, or in the very first divisions of the zygote.
Precisely. The parents do not carry the variation, but the child does. When a clinician identifies a de novo structural variation, particularly a massive deletion, a missense mutation, or a mutation that truncates a protein early in its synthesis, it acts fundamentally differently than a common SMP. A de novo mutation does not act like a drop in a bucket. It acts like a sledgehammer taken directly to the developing neural network.
The effect size is massive.
Huge. In fact, large-scale sequencing studies have demonstrated that de novo mutations, these spontaneous genetic sledgehammers, account for well over 10% of all diagnosed cases of severe autism spectrum disorder. So the clinical reasoning paradigm must hold two truths simultaneously. Common variants create a subtle polygenic risk background over generations, while rare de novo variants can catastrophically disrupt early neural development in a single generation.
That distinction brings immense clarity to how we should interpret genetic panels in the clinic. Now, while much of this multi-omics data is still in the research phase regarding diagnosis, there is one application of genetics that is already heavily utilized in daily psychiatric practice. We must discuss pharmacogenomics.
Yes. While we still struggle to utilize genetics to predict who will develop major depression, we are becoming exceptionally proficient at using genetics to predict how a patient's body will physically process the antidepressant medications we prescribe.
And this pharmacokinetic profiling primarily hinges on the cytochrome P450 enzyme system in the liver.
Exactly. The CYP450 enzymes are responsible for metabolizing the vast majority of psychotropic medications. The genes that code for these specific liver enzymes are highly prone to the exact structural variations, the CNVs we just discussed. Based on these structural duplications or deletions, a patient's metabolic capacity can vary wildly.
Let's walk through a clinical vignette to illustrate this. You have a patient presenting with major depressive disorder, and you prescribe a standard starting dose of an SSRI, let's say sertraline. 4 weeks later, they return, and their depressive symptoms are completely unchanged. The medication appears entirely ineffective.
Historically, the clinician might assume the patient has treatment-resistant depression, or perhaps they are non-compliant. But a pharmacogenomic swab might reveal that the patient possesses multiple duplicated copies of the CYP2C19 gene. They are what we classify as an ultra-ratchet metabolizer.
So they process it too quickly.
Yes, their liver is producing so much of the enzyme that it is actively destroying the sertraline before it can reach therapeutic concentrations in the central nervous system. The drug isn't failing. The dose is functionally subtherapeutic due to the patient's structural genetics.
The clinical utility of that knowledge is immense. You don't abandon the drug class, you simply adjust the dosing strategy. And the inverse scenario is equally dangerous, the poor metabolizer.
Yes. If a patient has deletions in the genes coding for CYP2D6, their liver lacks the enzymes required to clear certain medications. You prescribe a standard dose of a tricyclic antidepressant or a typical antipsychotic, and because they cannot metabolize it, the drug accumulates exponentially in their bloodstream. They rapidly develop severe, intolerable side effects or potentially toxicities like QTc prolongation from a dose that would be perfectly safe for the general population.
Pharmacogenomics effectively removes a massive variable of trial and error from psychiatric prescribing. It allows the clinician to predict efficacy and avoid adverse events before the first pill is even swallowed. It is the first true bridge toward personalized medicine in our specialty.
Indeed. It demonstrates that when we understand the biological mechanisms, we can intervene with absolute precision.
As we enter the final phase of our discussion, we must address the concept of integration. The overarching theme today is that the scale of this biological data, millions of SNPs, epigenetic tags, alternative splice variants, is cognitively overwhelming. A psychiatrist cannot memorize a trillion node network. How has the scientific field adapted to make sense of this complexity?
By fundamentally shifting away from reductionism and embracing systems biology. For a century, biology relied on a reductionist approach. Isolate a single gene, knock it out in a mouse, and observe what breaks. That approach is excellent for simple linear systems, but it fails entirely when applied to the human brain. Systems biology assumes that the whole is vastly more complex than the sum of its parts. We no longer search for the broken gene, we map the emergent properties of the entire biochemical pathway.
Emergent properties. Like consciousness itself, you cannot dissect a single pyramidal neuron and find consciousness inside it. Consciousness is a property that emerges only when billions of neurons interact in a specific pattern. Similarly, you cannot find the bipolar in a single genetic locus.
Exactly. To capture these emergent properties, we rely on massive bioinformatic tools capable of simultaneously analyzing millions of data points. A prime example is ChIP-seq technology.
You mentioned ChIP-seq in passing earlier. It stands for chromatin immunoprecipitation sequencing. How does this technique allow researchers to visualize the interacting network?
It provides a literal, real-time snapshot of the biological theater. In a ChIP-seq experiment, researchers use specific chemicals to freeze the live performance. They cross-link the transcription factors, the stage managers, permanently gluing them to the exact sequences of DNA they are interacting with at that precise microsecond.
Okay.
They then shatter the genome into millions of tiny fragments. Using highly specific antibodies, they pull out only the DNA fragments that have the transcription factor glued to them and sequence them.
So instead of just guessing where a transcription factor binds, you pull down every single binding site across the entire 3 billion letter genome simultaneously.
Exactly. It maps the entire regulatory web. And when you combine the data from thousands of ChIP-seq experiments with massive public databases like the ENCODE project.
The Encyclopedia of DNA Elements, which famously revealed that up to 75% of the human genome is transcriptionally active, identifying the millions of promoters, enhancers, and silencers hidden in the dark matter.
Yes. When you overlay the ENCODE regulatory maps with spatial expression data from resources like the Allen Brain Atlas, which provides a three-dimensional neuroanatomical map of exactly where and when these genes are turned on across the brain, you finally begin to see the matrix. You move from a static list of genes to a dynamic four-dimensional model of brain function.
So if I am a researcher applying systems biology, and I discover a patient has a mutation in one minor use exon of a seemingly obscure gene, I don't just stop there. I use these tools to trace the cascade I can deduce. This minor exon alteration changes the shape of a specific kinase protein. That misshapen kinase fails to phosphorylate a downstream transcription factor. Because the transcription factor remains inactive, it fails to bind to a vital enhancer element, which epigenetically silences a network of 50 downstream genes. Ultimately, that network failure results in a quantifiable drop in synaptic plasticity within the prefrontal cortex during a critical developmental window.
That specific, multi-layered causal chain is the holy grail of modern psychiatric research. It connects the microscopic genetic code directly to the macroscopic clinical phenotype. The profound takeaway for every clinician listening is this: mental illness is an emergent property. It is the catastrophic result of a network breakdown cascading across the genome, the transcriptome, and the proteome. And, crucially, that entire network is deeply influenced by the dimension of time, the developmental stages of the patient, and the psychosocial environment they are actively living in.
It is a breathtaking biological reality. To help solidify these complex models for our listeners, let's integrate some clinical reasoning. If you are a registrar sitting in a clinic right now or preparing for board examinations, how should you synthesize this information? I'm going to pose 5 clinical reasoning scenarios based on what we've discussed. Take a moment to think through the underlying mechanisms for each. You can pause the audio if you need a moment to think.
Scenario 1. A fundamental biological concept. You are reviewing comparative genetics. You note that while a simple organism possesses roughly the same number of protein-coding genes as a human, the human central nervous system possesses a vastly more complex, diverse array of functional protein networks. What specific molecular mechanism primarily drives this exponential increase in proteomic diversity?
The clinician must immediately recall the rehearsal space. The answer is the incredibly high rate of alternative RNA splicing, which occurs more frequently in the human brain than in any other tissue, allowing 20,000 genes to produce millions of distinct protein variations.
Scenario 2, focusing on the epigenetic mechanism. A researcher is analyzing a DNA sample from the postmortem brain tissue of a patient with severe early-onset schizophrenia. They identify a specific promoter region that is heavily marked by the histone tag H3K27ME3 alongside dense cytosine methylation at adjacent CpG sites. What functional state of chromatin does this biochemical signature represent?
Translating the alphanumeric code into physical structure, H3K27ME3 and heavy methylation are the ultimate molecular stop signs. The answer is that this signature represents dense, tightly packed heterochromatin, indicating that the genes in this region are heavily repressed and transcriptionally silenced.
Scenario 3, another deep mechanism. You are studying the rapid excitatory transmission governed by AMPA receptors in the cortex. You discovered that the mRNA transcript for the AMPA receptor contains a guanosine base, yet the underlying genomic DNA clearly codes for an adenosine base at that exact position. Which post-transcriptional process allows the human brain to alter the information content of an mRNA transcript without any mutation occurring in the DNA?
The rogue director with the red pen. The answer is ADAR-mediated RNA editing, specifically the deamination of adenosine to inosine, which the cellular machinery subsequently reads as guanosine, fundamentally fine-tuning the receptor's function.
Scenario 4. Focusing on clinical architecture. A patient with a strong family history of bipolar disorder undergoes comprehensive genomic sequencing. The report reveals absolutely no rare, highly penetrant single-gene mutations. Instead, the bioinformatics pipeline identifies thousands of minor, single-nucleotide polymorphisms scattered across multiple chromosomes. Which genetic architecture model best explains how this patient developed the disorder?
Moving away from Mendelian genetics to the bucket analogy. The answer is the common variant, common disease model, which posits that complex psychiatric traits arise from the cumulative polygenic burden of thousands of common sequence variations interacting with environmental triggers.
Scenario 5. A final clinical application. In evaluating a young child presenting with severe neurodevelopmental delay and autism spectrum disorder, a high-resolution chromosomal microarray reveals a massive microdeletion on chromosome 16. Further testing of both biological parents confirms that neither parent carries this deletion. What is the precise clinical term for this type of mutation, and how does its significance differ from a common SMP?
The genetic sledgehammer. The answer is a de novo structural mutation. It occurred spontaneously, carries a massive effect size with high penetrance, and is responsible for a significant proportion of severe early-onset neurodevelopmental disorders, unlike the subtle cumulative effect of common SNPs.
Excellent clinical synthesis. For listeners wanting to deeply review the biochemical pathways behind those scenarios, Chapter 18 of the textbook provides the exhaustive molecular detail. Mastering this material is not just academic trivia. It provides the fundamental vocabulary required to understand the future trajectory of psychiatric medicine.
We have covered an immense amount of ground today, from the physical winding of chromatin spools to the exponential maths of the proteome. But I want to leave you with a final, slightly provocative thought to reflect upon as you walk into the clinic tomorrow. We discussed the ALU elements, those 300 base pair retrotransposons, the jumping genes. We established the biological reality that these elements are actively waking up, transcribing themselves, and physically reinserting into new, random locations within the DNA of our individual neurons as we live our lives. The stress we endure, the memories we form, may subtly influence how frequently these elements jump. If our neurons are constantly physically altering their own underlying code, is there truly one single static genome that defines who you are? Or is the reality far more complex? Is your brain, in fact, a magnificent mosaic composed of billions of unique, ever-changing genetic identities, a physical network that is literally being sculpted, edited, and rewritten by your daily experiences?
It is a profound, paradigm-shifting realization. Your biology is not a cage that traps you. It is a dynamic canvas that continuously responds to your life.
Here are the details of this episode.
Genome, transcriptome, and proteome. The molecular genetics, biochemistry, and multi-omics underlying the neurobiology of mental disorders. Byline. Layers of instruction from genetic code to biological expression and psychiatric vulnerability.